Hello, everyone, and welcome to today's AAPS eChalk Talk. Today's eChalk Talk is hosted by Celerion. You may submit your questions at any time by entering them into the Q&A box of the Zoom platform. All questions will be answered at the end of the program. It is now my pleasure to introduce today's speaker, Dr. Michael Gröschl. He is Technical Director for Automation and Computerized System Validation at Celerion. Michael, the floor is yours.
Thank you very much. So, ladies and gentlemen, I have the pleasure to speak in this webinar today on the topic of saliva as a reliable sample matrix in bioanalytics. I would like to provide a brief insight into the physiology of saliva, its diverse biological functions, and the formation and transfer of various molecules into saliva. Afterwards, we will discuss the possibilities of saliva collection, as a reliability of the subsequent analysis crucially depends on this. The majority of the talk will then be devoted to the presentation of application examples from various fields of bioanalytics. I have been involved in saliva analysis, meanwhile, for a quarter of a century, mostly in the context of endocrinology and behavioral research. So there's quite a number of publications on this topic, which interested listeners are welcome to request by email at any time. Now to the actual lecture. Saliva fulfills a multitude of physiological functions. In addition to preserving the teeth through its buffering properties and maintaining healthy tooth enamel by providing the necessary minerals, its primary role is in food intake. Without saliva, swallowing would not be possible, nor would be pre-digestion by enzymes like amylase. And we would also have no sense of taste. But saliva plays also an important role in the immune system. The mucous membranes of the oropharynx are constantly fighting against viruses, bacteria, and fungi. And the antibiotic and antiviral enzymes and peptides in saliva form a first highly effective line of defense. Anything intercepted here cannot enter the bloodstream and cause infections and diseases. This is the main reason why wound licking in animals is such a successful way to clean and disinfect their wounds. Saliva is a mixture of secretions from various glands located in the oropharynx. A distinction is made between the more watery saliva, which is primarily secreted by the parotid glands, and the more viscous, sticky secretions of the other major salivary glands, being the sublingual, the submandibular, and the tubarial glands. Interestingly, these tubarial glands were discovered and first described in 2020. A multitude of tiny glands located throughout the oral mucosa are responsible for maintaining the general moisture content of the oral cavity. The content of biologically active substances can vary considerably depending on the composition of saliva. In general, it can be said that anything you can detect in blood should also be detectable in saliva. However, this should depends on the transport mechanism. With simple diffusion, as in the case of unconjugated steroids, for example, we often find a 1:1 ratio between the levels in blood and saliva. However, as soon as active transport mechanisms come into play, the situation becomes more complex. In some cases, we find much lower concentrations. In others, interestingly, even significantly higher levels, indicated a targeted transport with a physiological function. Not to be forgotten, there are also substances that are produced and secreted autonomously in the salivary glands, which are usually directly related to the primary functions of saliva in digestion and wound healing. Now let's move to saliva collection. Here only a few examples shown. When I first conducted my own research on this topic, there were almost no commercial collection systems available. They came on the market around the year 2000 and have now developed a wide variety. Therefore, before starting a study, you should consider which collection system would suit best for the analyte you want to test. A major factor is whether the system could hinder or falsify the analysis, or whether with especially with adsorption modules, the analyte can be completely recovered during the sample processing. I published several papers on this topic at the time, which demonstrated the complete inadequacy of saliva collected on cellulose or cotton rolls for the analysis of cortisol and insulin. As a result, substitute products such as widely used polyester-based salivate, shown on bottom right, came on the market. With this material, almost 100% of analyte recovery could be demonstrated. The use of systems with integrated stabilizer solutions, as exemplarily shown in the bottom left, is recommended for later PCR analysis. One key advantage of saliva collection compared to blood sampling is its simple and non-invasive way of sample collection, which can be performed by non-medical personnel, even by the patient themselves, even in areas far away from medical facilities. Saliva analysis is particularly accepted in pediatrics and increasingly also in geriatrics, where blood sampling is technically challenging. But also, saliva is also interesting and increasingly being used in behavioral research and reproductive medicine in zoological gardens. Bottom right, an image from my own diploma thesis on steroid determination in guinea pigs during pregnancy. While emphasizing the advantages of saliva as a bioanalytical sample matrix, for the sake of balance, it's also necessary to mention the disadvantages. First, saliva can be quite viscous, sticky, especially if collected improperly and also in some diseases, such as in Sjogren's syndrome, which severely limits the subsequent processing of the sample in the laboratory. Other disadvantages are also related to the physiology of saliva production. The often lower concentrations require often very sensitive analytical methods. But fortunately, much has been achieved related to analytical sensitivity in the recent years. Lower concentrations often result from the fact that some substances in the blood are mostly bound to large transport proteins or binding proteins, but only the unbound and freely available, and therefore also the physiological active portion, can pass from the blood through the barriers, the membranes, into the saliva. This makes saliva of particular interest to those researchers who are investigating the biological active portion of the substance. Most important, however, is to adhere to a standardized procedure for saliva collection to avoid contamination, and also to consider the influence of varying secretion, for example, due to circadian rhythms. Now let's move on to the application examples. Saliva analysis is most widely used in endocrinology and related fields such as in behavioral research. However, the detection of salivary biomarkers for oral and gastrointestinal health, as well as for systemic and chronic diseases, is also increasingly developed. While the use of saliva as a sample matrix in pharmacokinetic and pharmacodynamic studies is still in its infancy, its application in forensics and sports medicine is already well advanced. Now for the specific examples. The classic example is the detection of small hormones, such as melatonin from the pineal gland which weighs only about 230 Dalton, or steroids, which weigh around 300 to 350 Dalton. This figure shows typical circadian pattern of melatonin and changes of salivary progesterone concentrations during the menstrual cycle. You can clearly see the increase in melatonin during the dark phase, also during night, as well as the extreme increase in progesterone during the luteal phase of the menstrual cycle. However, as soon as the topic of steroids comes up, the conversation, from my knowledge, immediately turns to wellness assessment. Some manufacturers have taken this as an opportunity to release saliva hormone kits for home use. Personally, I'm not convinced of the value of these data obtained with such methods, as any single point determination ignores the individual influences on sampling and circadianity, and the assessment of health, or let's say the menopausal status of a woman, using such data is at least questionable. Next, let's turn to biomarkers of gastrointestinal tract. I've listed a few examples here. First, there are peptides of the non-specific immune defense in the oropharynx, such as defensins, which, as the name suggests, have a strong antibacterial effect. Small peptides such as catalycidin and adrenomedulin have also antibacterial effects, most likely due to their high charge, which blocks the ion channels of the bacterial cell membranes. So to speak, these are like protein-based cannonballs to the bacteria. In addition to the antibacterial peptides, the rapid wound closure of the mucosa induced by growth factors present in saliva, also the so-called cytokines, is also crucial for fighting infections or preventing infections. In addition to EGF, VEGF, FGF7, my favorite peptide, leptin, should also be mentioned here, whose proliferation promoting effect on mucosal cells I was able to demonstrate for the first time. Of particular interest here is the cytokine cascade induced by the increase of salivary leptin. This finding induced further studies which ultimately demonstrated that leptin in saliva can also be used as a reliable biomarker for specific tumors of the salivary glands, as well as the squamous epithelia. Other proven tumor markers in saliva include, for example, telomerase activity and various interleukins. Next, a few examples of therapy monitoring for systemic diseases, which are also related, of course, to biomarkers. Specific shifts in the concentrations of certain peptides and proteins in saliva have been observed for both cystic fibrosis and diabetes. And these changing concentrations are used for monitoring during drug therapy. It should also be noted, of course, that due to the shift in the ion balance, saliva production, especially in cystic fibrosis, deviates significantly from the norm. Saliva has so far only rarely been used in vaccine studies, even though, as in the example shown here, an increase in specific IgG antibodies against a meningococcal vaccine can be detected in saliva. This is merely a side note. Because the central immune globulin on the mucous membranes, as well as in saliva, is secretory IgA, which, as we know, serves more as a non-specific rather than a specific immune response. And in general, IgG, which is predominantly present in the bloodstream, the concentrations in saliva are so low that a reliable analysis is very unlikely to have a future, to be honest. The situation is completely different with therapeutic drug monitoring. The example on the left shows the cause and the correlation of the cytostatic drug busulfan in plasma and saliva in children with leukemia. This study was conducted years ago in the Clinic for Children and Adolescents at the University of Erlangen, where the patients were treated and a target level of the cytostatic drug was to be set. Considering that these poor children are marked by the disease and weakened by the cytostatic therapy, at least replacing blood sampling with a much more gentler saliva collection seems to be a small relief to them. On the right, we see the correlation of indinavir, a protease inhibitor used in HIV infections, in plasma versus saliva, as in matched samples. Despite the significantly lower saliva concentrations, there is, same as in the busulfan, a high correlation between the two matrices. So indicating that the saliva can be taken as an alternative to the blood collection. The detection of narcotics, of drugs of abuse from saliva has found its way into forensics, forensic medicine and law enforcement, and this surprisingly quickly. As a rapid alternative to the previously common urine test, which understandably poses certain challenges when conducted on the roadside, the detection of opiates, cocaine, and other drugs of abuse from saliva is a fully established alternative meanwhile. This now leads to the final area of applications I want to present, sports medicine. Here too, the rapid detection from urine was long considered the gold standard. But respecting the athlete's privacy, unfortunately, also created some opportunities for manipulating the sample behind the curtain. So saliva, on the other hand, can be collected under direct supervision, as shown here in the picture, significantly limiting the potential of fraud. The World Anti-Doping Agency, the WADA, has meanwhile fully approved saliva as a matrix for rapid doping tests. So let's summarize. Saliva is now a widely accepted sample matrix in various biomedical and bioanalytical applications. The collection is quick, it's non-invasive for the patient, and does not affect the concentration of the analyte. Maybe, for example, due to the stressing blood collection. Especially when repeated collections are required, also short intervals or very close profiles, saliva clearly has an advantage over blood collection. And it's also very helpful in screening applications. In general, however, you should never ignore the specific physiology of saliva formation, the expected mostly lower concentrations, and the need of highly standardized collection regime. At the very end, coming already to the end now, some gems from the media, mostly in German, sorry for that, ranging from print media via radio and television, even up to the Wall Street Journal, which also examined that topic from an economic point of view. So how saliva testing could be economically beneficial. So now I thank you very much for your attention, and I'm happy to hopefully answer your questions. Thank you very much.
Thank you so much, Dr. Gröschl. We do have a couple of questions. The first question is, Which type of saliva is preferred for analysis, stimulated or non-stimulated?
Well, to have a very standardized flow, it's preferred to stimulate the saliva a little bit, preferably to rinse the mouth before the collection with water, spit this out, and then stimulate the saliva flow by chewing, which is already integrated in some devices like this salivette, for example, where you chew a little bit and then stimulate and have a constant saliva flow. So this is preferred compared to passive spitting or drooling into a collection device.
Great. Another question, how is the stability of saliva?
Stability of saliva is good enough to collect the sample at home, outpatient sampling for, let's say, therapy control, but not good enough to keep it there for weeks. Also the normal way to handle this, let's say in pediatrics, is the people collect a typical profile, morning, noon, evening collection, keep these samples in the fridge and ship it then by mail at room temperature, so to say, to the clinic. And this is specifically for steroids.
Great. Another, do you need to know or deeply explore the plasma protein binding in order to justify the use of saliva PK as a surrogate for plasma PK?
When the analyte is not known to me, I would strongly recommend to have a short correlation between the plasma and the saliva concentrations to get a rough estimate.
Great. Would saliva be a good matrix for neurological disease, for example, concussions, AD or PD? And if so, are there any examples?
This is, in fact, a question that I cannot answer right now. There I would have to go into the literature first. Sorry.
Okay. That's great. Does saliva really have a future in bioanalytical studies?
Yes. Yes, it depends on the analytical parameter. Again, some are proven, nicely published, so there everything is clear. For others, unknown drugs, if you want to run a PK study for, with your drug on saliva, of course you have first to see, can I recover the drug completely from the collection device? How is how does the profile look like? Is it in the same? Does it appear in the same time intervals as in plasma? Is it equivalent, or do I have to consider some differences? But yes, it has definitely a future when you do some homework up front.
Thank you. Is there any procedure to reduce the stickiness of saliva before analysis?
Freeze thaw.
I'm sorry, say that again.
Oh, freeze and thaw the sample, and centrifugation. Then the mucins and the cell debris goes down, and you can work with a clear liquid.
Okay, great. How is the matrix effect on saliva sample recovery?
That's a good question. How's the matrix effect? Honestly, I do not have a instant answer on this.
We have you stumped.
Can happen.
That's great. Can we collect exosomes for RNA sequencing from saliva?
Yes.
Great. And we have seen saliva samples that initially inhibit qPCR, but did not after going through a freeze thaw. Is this anecdotal or is there something to it?
Might have to do with binding or how to say, the inhibition of the analysis by components that are surrounding your analyte, but which then, as I said, which you can get rid of by freeze thaw.
Okay. I think we have time for another question or two. Is a single freeze thaw cycle sufficient to reduce stickiness and viscosity?
Depends on the analyte. Sorry, that is best answer on this question. It depends on the analyte. I would recommend to have during your method development such recovery experiments included to see how many cycles are required to achieve an almost 100% recovery.
Great. Well, it looks like that was the last question. Dr. Gröschl, we really want to thank you so much for coming and speaking with us.
Thank you.
And again, thank you to Celerion for partnering with us. And we look forward to seeing everyone again at a future eChalk Talk. Thank you so much and have a wonderful day.
Same to you. Thank you very much.